Welcome to our final video on lipid metabolism, and in this video we're going to focus in on membrane and storage lipids and cholesterol. So if we take a look at our metabolic map, we're really looking at these two areas in orange and blue, and I've blown them up a little bit right here. So the orange are the membrane and storage lipids, and the blue is the cholesterol. So if we take a look at this, let's highlight some things. So up here, here's our kind of membrane and storage lipids that we're going to talk about synthesizing. And the starting material for that is right here, this phosphatidate. If you backtrack from that, we can see that we kind of came at it from this glycerol 3-phosphate, from the glyceraldehyde, so eventually back from these carbohydrates, right? We ended up making this phosphatidate, which we're going to consider this starting point for our discussions today. All right, and we're also going to talk just a little bit about these. So here's the triacylglycerols, those triglycerides. Okay, and then thinking about separately cholesterol over here in the blue box. So we have cholesterol right here. And if I want to think about my starting material there, I'm going to have this beta-hydroxy, beta-methyl, glutarol, CoA. And it's going to turn out that similar to other processes we've seen before, we have to activate that to this mevalonate, I'll get it, for it to form that cholesterol. All right, from the cholesterol, we can synthesize various steroids, and we can synthesize bile salts or bile acids. All right, let's take a look. We're going to start up here, okay? And we're going to talk about that phosphatidate that we said was really our starting point for these discussions. So it is a precursor of those storage lipids and many membrane lipids. And it's formed from essentially this glycerol 3-phosphate. You'll remember I highlighted that up there. And we're going to add 2 fatty acid chains to it. All right, and you can see those, right? We've got 1. So those are pulled off these kind of acyl-CoA molecules. We've got 1 added here. We've got 1 added here to that glycerol piece. All right, and then here's that phosphate. Okay, so we've got our phosphatidate. 2 fatty acid chains, glycerol backbone in black, phosphate in the pink. That phosphatidate, so right here, is the precursor for a ton of different things. It's a central metabolite that we kind of have at the ready to be modified depending on what the cell needs. It can be made into many things. If you study this more, you'll hear it talk about it being active versus inactive. So when it's active, it'll kind of come over this way and it'll produce these types of things, including, notice, our triacylglycerols. Whoops. When it's inactive, and we're not going to talk about details of when it is and isn't, but it'll come back this other direction, right, and produce other molecules that are needed for our body. So the details on how this is regulated to form different lipids is not nearly as well understood as fatty acid metabolism. It's really an active area of study. But we do know that many of these lipids are synthesized in the liver. We know that a key regulatory enzyme is this phosphatase. Okay, so phosphatidic acid phosphatase, a regulatory enzyme. So phosphatase would clip a phosphate off. The kinase going back the other way would add the phosphate back on. All right, and if we look, this is interesting. So loss of this phosphatase activity in mice results in loss of body fat and development of insulin resistance. So there's something important about this enzyme for our lipid metabolism that's not fully understood yet. But if we could control development of these triacylglycerols, we could maybe use that as some sort of treatment for obesity. All right, we're going to skip over now and talk about cholesterol. So cholesterol can be synthesized in many tissues, but the primary site is the liver. Okay, so liver is our primary site of cholesterol synthesis. Cholesterol we know is critical for membrane fluidity, plus for starting materials for these steroids, for these bile acids. Cholesterol synthesis occurs in 3 stages, and we're going to look at each of these 3 stages in a little bit more detail. And the reason we care about how it's synthesized is that if we understand how it's synthesized, we can better understand how to treat high cholesterol. The processes that we go through to get from our starting materials to this really complex organic molecule are pretty impressive. So we're going to take a look. So the first stage, remember 3 stages. First stage is the synthesis of that, essentially that activated molecule. Okay? So to get to this mevalonate is a committed process. Once we do this, now it's going to move on and make that cholesterol. This is the regulatory step. Okay, so this is our regulatory enzyme. Few things to notice. This is a reductase. Okay, so it's causing our molecule over here to be reduced. Notice we have shorthanded that, the hydroxymethylglutarol, HMG, CoA, and then it's a reductase because we're reducing that. Because we are synthesizing molecules, we know that instead of NADH, we'll use the NADPH, and you see that here. So that's being oxidized, giving away its electrons. Okay, so essentially we're spitting off this coenzyme A to give us the activated molecule. And again, we care about this, particularly about this regulatory enzyme, because high cholesterol is essentially a disease state. And if we understand the regulatory enzyme, we might be able to better find drugs that will help work against that disease state. All right, continuing on, still in stage 1. Okay, this activated kind of starting material uses a bunch of ATP and steps through this process to get over here to this 3-isopentenyl pyrophosphate. Think pyrophosphate, remember that's 2 phosphates bound together, and we see that right here. You do not need to memorize the details of this, okay? What I want you to realize is that, wow, there's quite a bit of synthesis going on here, right, as we go from stage to stage. And it's going to get even more impressive as we move into stage 2. Okay, so in stage 2, we're going to synthesize squalene, which has 30 carbons. Okay, and to do that, we're going to use 6 molecules of that product from stage 1. Okay, that isopentenyl pyrophosphate. So each one of those has 5 carbons, so we need 6 of them. If we look at the steps, we're essentially combining these 2 molecules to give us this one. And then we're tacking on another one of these. And now we've made this. If we've done this whole series again, we've got 2 of these to combine to make the squalene. Again, you don't need to know the enzymes that are associated with this or these specific steps. you should just be impressed. Okay, cholesterol is a large, complex molecule. If we go back in our metabolic pathway from before this, you'll see that eventually we'll get back to acetyl-CoA. So we're taking this really small piece and developing up till now this squalene, this 30 carbon structure. And then in stage 3, we're going to introduce the ring structure. All right. This stage will end with our cholesterol over here. So we start with this 30 carbon hydrocarbon. We go through a bunch of steps. Y'all notice there are 19 more steps here that we're not even showing. It is a really long synthetic route. It takes energy to get there. Again, you don't need to know all these specific molecules or anything. I just want you to appreciate that your body goes through a lot of work, all right, to make this cholesterol. What we really want to talk about is regulation of synthesizing that cholesterol, because if we can regulate it, that gives us potential to help people who have high cholesterol. So again, liver is the major site of cholesterol synthesis. Okay, can be synthesized in other cells, but liver's the major site. Regulation of cholesterol synthesis occurs by controlling that regulatory enzyme I pointed out earlier. Okay, that was the enzyme that kind of activated our starting material. Okay, it gave us that mevalonate. And there are several ways this enzyme can be regulated. So cholesterol is really important to us, so it makes sense that our body would have multiple ways to kind of try to keep it at the right levels. All right, so we're looking at regulating this enzyme. There are other ways actually to regulate cholesterol synthesis, but we're going to focus in on this one. And it can be regulated in 4 different ways. One is the level of transcription. Okay, we can stimulate the synthesis of the mRNA needed to synthesize the protein, to synthesize this enzyme. So we can regulate it all the way back at the level of transcription. Essentially turn the gene on and off, right, to synthesize this mRNA. We can regulate at the level of translation. Okay, that if we start building up this mevalonate or this dietary cholesterol, that will help control then this translation using that mRNA to make the protein. All right, so we'll push it forward or backwards depending on the level of these various metabolites. All right, then we can keep going. The third way is once we already have that enzyme, we can degrade it. All right, so we can essentially destroy our enzyme. So it gets acted on by a protease, and that can often be controlled by cholesterol concentrations or levels. And then the other thing is, we've seen this over and over, right? Phosphorylation. So that enzyme could be phosphorylated. And let's think about this. If it's going to be phosphorylated by AMP-dependent kinase. So when the energy charge is low, it's going to phosphorylate the enzyme. Okay, and if it's phosphorylating the enzyme, we can think about that. If the energy charge is low, are you going to want to be using your energy to synthesize cholesterol? Probably not, right? So if our energy charge is low, we got a lot of AMP. We're going to activate this kinase that will phosphorylate our enzyme, and that will inactivate our enzyme. Okay, it will keep it from activating. It won't make that starting material, right, the mevalonate, and so we won't keep making our cholesterol. And this is really important, right, because if you think about it, if our energy charge is low, eventually those carbons, if you trace them back, are coming from that acetyl-CoA. We're going to want that stuff in our citric acid cycle at that point. All right, so things that can control this. All right, quick check for understanding here. Outline mechanisms of regulation of cholesterol biosynthesis. All right, so what you should know at this point. Cholesterol has a complicated synthetic pathway. You do not need to know the all the individual enzymes, metabolites, all of those, but you do need to be appreciative of this complicated pathway, and you need to know that there are multiple ways to regulate it. You should be able to write down the name of the regulatory enzyme. So remember, that's the one we've been discussing. You should have an idea of where in cholesterol synthesis this enzyme functions, which remember was at the very beginning. Okay, and then you should understand that it can be regulated in 4 different ways, right? We can regulate it at the transcription phase. We can regulate it at the translation phase. We can regulate it by digesting it, right? Proteolytic digestion of the enzyme. Okay. We can control it by phosphorylation. Okay, and that would be from our kinase cascade, right? From hormone signaling, turning on that AMP kinase and phosphorylating it. Okay, why do we want to know these things? Because, again, high cholesterol is a problem, and the more we know about the synthesis, the more we can target drugs at these places of regulation. So let's look a little bit at how lipids and cholesterol move around your body. So lipids can be consumed in the diet or synthesized mostly in the liver, but we need them to be able to move around the body. Lipids are not particularly water soluble, right? They're not going to be, so they're going to need to be packaged to move around. We looked earlier at these chylomicrons, and then we reviewed it in a recent video. Okay, so remember that these chylomicrons are lipids packed up in this lipoprotein. Okay, they are this is a specific type of a lipoprotein. And these are the ones that are coming kind of from your diet. When they're synthesized in the liver, they also need to be packaged in these lipoproteins. But the lipoproteins that we see in the liver are different from those chylomicrons. They have different proteins on the surface that can be recognized by cell. They're a bit more sophisticated. So over here at the right, we have a cartoon image of a low density lipoprotein. We're going to talk about that more in just a minute. But on the surface, okay, you can see these proteins. So these would be proteins that would be recognized by certain receptors on various cells. Okay, the green and pink here, we've got our phospholipids kind of coating the surface here. And in the middle, we've got our cholesterol. And that cholesterol is stored as a cholesterol ester, so kind of similar to our triacylglycerol as an ester. And so it exists in kind of just a droplet form. Remember the triacylglycerols were in kind of that random droplet lack of organization? Same thing here. All right, let's talk about how those lipoproteins transport the cholesterol and triacylglycerols throughout the organism. All right, so we just said that, hey, we're going to transport them in the blood through these lipoprotein particles. Those are going to consist of protein component and various lipids, depending on the type of particle. The proteins serve to solubilize the lipids and to direct the particles to specific targets. So lipoprotein particles are classified according to density. The greater the proportion of lipid, the less dense the molecule. Let's think about that. Okay, as we're talking about density, it has to do with the ratio of our triacylglycerols to our cholesterol. All right, triacylglycerols are less dense, so the higher the proportion of triacylglycerols, the less dense our overall lipoprotein. Okay? Low density lipoprotein is the major carrier of cholesterol in the blood. And then the high density lipoprotein essentially carries cholesterol released in the blood back to the liver. So through reverse cholesterol transport. And we're going to look at that over these next couple slides. So let's take a look at just some properties of lipoproteins. So we have density right here. We have our triacylglycerols right here. We're not going to worry about these as much, but I'm just going to cholesterol ester, just cholesterol. This is our phospholipid. And this is protein. Okay, and over here on the right, we're essentially looking at the percentage that is each of these. So we said the density had to do with the ratio, right, of triacylglycerol to cholesterol. And let's see if that holds true over here. So as I look at my chylomicron, I've got a lot of the triacylglycerol. I have a whole lot less cholesterol. And we said the triacylglycerol are less dense. So the more of them I have, the lower my density. And we can see that this does indeed have a pretty low density. So these are actually the least dense. When we go down and look at the ones that are most dense, those are our high density lipoproteins. And we can see that indeed, I've got a whole lot less of our triacylglycerols, and I have a whole lot more cholesterol. And so those are more dense. In the middle, these three are going to be the three that carry lipids from liver to the cell. So they carry the cholesterol and lipids from the liver to the cell. All right, and if you think about it, and I'm trying to remember if I have I do have a little picture of it. We'll talk about it. As the very low density lipoprotein moves through the bloodstream, lipases will act on it. They'll break off those fatty acid chains. So we've got triacylglycerols, right, in this. But as it moves through the bloodstream, lipases will break off some of those fatty acids, and those fatty acids will diffuse out of the lipoprotein into cells. And as we do that, we have less and less and less of this left in our cell, or in our lipoprotein. And so as we have less and less of this, our density starts going up. Okay, and so we move from the very low density to the intermediate low density and finally to the low density lipoprotein. Okay. All right. So just to put this in here too, this high density was bringing the cholesterol back from the cell to the liver. So let's look at kind of a cartoon of this process. So bottom left, right here, we have your liver. All right. And in this cartoon drawing, we can think about the fact that the liver can produce these bile acids. So it uses cholesterol to make bile acids and takes them up to the small intestine eventually. And that was where, when we did that first video on digestion, we talked about those bile acids being used to homogenize fats so they can be acted on by the lipases and make their way into these chylomicrons. Okay, so bile acids can be taken, and then they can also be returned from the large intestine. Okay, but in the intestine, those dietary fats and cholesterols are changed into chylomicrons, or packaged. All right, and then they get broken up. We get those free fatty acids released by the action of lipases, which can be stored in fat cells, can be taken to things like muscle cells. Okay, and then eventually they can come back into the liver to be used as energy or repackaged as triacylglycerols. Okay, so that's kind of the path for our dietary fats and cholesterol. If we want to look at our path for the cholesterol that's synthesized in the liver, it leaves as that very low density lipoprotein. It's got a decent amount of the triacylglycerols. As it moves around, the lipases work on those triacylglycerols, and some of those fatty acids leave, right? And they also go to fat tissue, they go to various cells to be used. And so now, though, we've got a higher proportion of cholesterol because it's still all in there. Only the triacylglycerols are getting broken up. And as we keep going, it keeps happening, and we've got even a higher proportion of cholesterol until we get to that LDL part. And now we have the right ratio of proteins at the surface to the cholesterol to undergo endocytosis, which is a process that pulls that cholesterol into the cells. So if we look at that, it's the low density lipoproteins, okay, that can deliver cholesterol to peripheral tissues. And at the tissues, cholesterol enters by this endocytosis. 3 steps. Start with the green one down here. All right. LDL is going to bind to a receptor, an LDL receptor at the surface. So here we have our lipoprotein, and here we have that receptor. Okay, and then go into step 2. This is going to form this receptor LDL complex that's going to get pulled in and internalized. So it gets pulled in. This is different than with our triacylglycerols, right? For those, fatty acids are snipped off and make their way into cells individually. But cholesterol is getting pulled in as a whole vesicle full of it. So that was step 2. There's our endocytosis. And then in step 3, that LDL is going to be hydrolyzed, okay, in lysosomes. And so we're going to break apart, we're going to get amino acids, we're going to get the cholesterol, and this LDL receptor molecule will return to the surface so it can grab another one of these. So understanding these processes is going to be important for thinking about how we can help people that have high cholesterol. So let's look at a couple examples of that. So in an inherited disease, okay, this familial hypercholesterolemia, it is a genetic disease that results from the absence of a functional LDL receptor. So go back up here. Doesn't have the receptor, can't grab that cholesterol from the bloodstream, those lipoproteins. And so studies of people with this disease who have definite problems with cholesterol have given us a lot more information for more common problems with cholesterol. People with this have excess cholesterol that collects in various tissues of the body because it can't get pulled into the cells and used, so it's kind of more floating around there. Excess LDL becomes oxidized, and the oxidized LDL is ingested by these macrophages. And so much of this stuff is present that the detergent properties of the cholesterol cause the cells to appear foamy. And these kind of foam cells become trapped in blood vessels and contribute to the development of these precipitates and hardening of the arteries. Okay, so left here would be normal, right would be disease, where you can see this plaque forming. So one thing this tells us is that if we can control these LDL receptors, we can maybe pull more of this cholesterol out of the bloodstream and help prevent this from happening. Now, if we're thinking that way, hey, we want to be able to control these receptors, then we need to know how this cycle of pulling this in and then sending that receptor back is regulated. And it turns out there's a specific protein, you don't need to remember the name of it, but here it is, that's secreted by the liver that binds to the LDL receptor, okay, and prevents it from recycling. So it keeps it, right? This protein would keep, that's not what I wanted, would keep this from happening. And then eventually this receptor gets degraded and it's no longer there. So this protein is not a friend to us being able to pull more cholesterol from our bloodstream. So if we could make that not function well, okay, if we had a loss of that function, that would mean we could pull more of this in, right? Because it's going to get pulled in, and then it is going to be able to be recycled and come up here so we can pull in another one. Okay, so a drug that blocks that protein could help reduce high blood levels of cholesterol. So it's one way we can use our knowledge of the biochemistry to think about solutions to health problems. As we're talking about arteriosclerosis, HDL seems to protect against it. In the process of reverse cholesterol transport, going out and grabbing cholesterol that was left over from cell death and things like that and pulling it back to the liver. HDL removes cholesterol from those macrophages and returns it to the liver. Okay. We also know that HDL associates with an enzyme that may help degrade some of that oxidized LDL. So, and we know that ideally we want to increase our HDL levels and decrease our LDL levels for general health. Okay, let's look at another way that we could help manage cholesterol levels using our biochemistry knowledge. So another strategy is this two-prong approach. We want cholesterol out of the blood. We know that our bile acids are made from cholesterol. So if we could essentially make our body make more of those bile acids, we'd be using up that cholesterol. All right, we're going to do that, and we'll look at this on the next slide. By ingesting a resin that binds to the bile salts, and we're going to talk about that in just a second. And then another piece of this two-prong approach, okay, is inhibiting that regulatory enzyme, okay? And these are what you hear called statins. Here's an example of one. And remember our discussion of competitive and non-competitive inhibitors. All right, so this is a competitive inhibitor for the active site, okay, on our enzyme. And so if we have that in there, our HMG-CoA molecule can't get in there, and we won't make our activated starting material. Okay, and so hence that will slow down our processes of forming this cholesterol. Couple other things just to go along with this that actually I'll mention one of them in the next slide. Let me look just at my notes, make sure, because this stuff's interesting, right? You hear about these things. You hear about statins. All right, let's take a look at the next slide. I think that's where we want to go next. All right, so if cholesterol is a precursor to steroid hormones, vitamin D, and bile salts. This is the part we're interested in here. And those bile salts are detergents that we used in the intestine, right, to make those lipids more accessible so they could be digested. So your liver made the bile salts from the cholesterol. It uses up cholesterol to make these bile salts, ships them off. They get stored in the gallbladder until they're needed, and then they're secreted into the small intestine. Now, it turns out that these bile salts can essentially be recycled through, and we mentioned that earlier. Like you can recycle them back through and use them again. But if we want to use more cholesterol, we don't want to be that efficient. So essentially, we can ingest something that will bind to those bile salts, okay? When that happens, they can't get recycled. They'll be excreted in the feces, and your body will have to make new ones, okay? And so it'll use up more cholesterol. All right. So the most important pieces out of this chapter is really understanding how cholesterol is synthesized, or out of this segment, we summarized the earlier piece. Understanding how cholesterol is synthesized, not the details, okay? But you should understand the regulatory enzyme. You should understand how that regulatory enzyme can be controlled. You should understand how cholesterol is taken up into the cells, and then think about how that helps us determine and understand how drugs work to help with these cholesterol levels. All right, that is it